All right, so thank you everyone for joining us. And today we're very excited to welcome Dr. Bill Hlavacek from Los Alamos National Laboratory to hear about his new tools for parameter optimization of biomodels, especially for incorporating some data from some qualitative data for these models. So let's welcome Dr. Bill Hlavacek.
Okay, let me share my screen. Can everyone see my slide and hear me?
Yeah.
Am I good to go?
Mm-hmm.
Yes.
All right, so I'm happy to be here and share our work with you. I'm going to talk about a proof of principle demonstration and then briefly introduce software that is general purpose and that enables this approach. The work that I'm going to talk about is mostly the work of a postdoc who's here with me, Ishan Mitra. Ishan will help me with questions that I can't answer, I hope. So before getting started, I just want to acknowledge the people involved. Again, Ishan did most of the work. My group collaborates with an old friend, Rich Posner at Northern Arizona on software development, and Herbert's group helped us with support for LibRoadrunner in the software that I'll mention at the end. We have funding from NIH, and there's a paper about the work that I'll talk about today, and just recently we released the source code for the software that I'll introduce. So the work is really inspired by the work that John Tyson and coworkers have done. This is a slide showing one of their first papers where they took this approach, and I've highlighted a part of the abstract. And I'll just quote from it. So it says, the budding yeast cell cycle is constrained by the phenotypic properties of more than 100 genetically engineered strains. So they're just saying that we know how the cell cycle regulatory system behaves through mutants. And then they go on to say, we show that a mathematical model built on a consensus picture of this control system is largely successful in explaining the phenotypes of mutants described so far. So they've built a model that's consistent with what we know about the phenotypes. And interestingly, what we know is largely qualitative. And so they parameterize this model by taking advantage of qualitative data, which is very unusual, I think. Now, just a couple of limitations of what they've done. They parameterize their models in this pioneering study and then in later work through hand tuning. So that's laborious, and in part for that reason, it's somewhat difficult to reproduce. And they've focused on these qualitative observations about mutant viability, and there's been little consideration of quantitative data. So my goals for today are to explain how we automated the approach that Tyson Group has used to parameterize their models for cell cycle control, explaining how this approach can account for qualitative data and also any available quantitative data that you might have. And then I just want to briefly introduce the software that we've developed that enables this approach. So years ago, I was on a panel with a bunch of modelers, and an experimentalist in the audience asked, what could experimentalists do to help you with your modeling work? And the first guy said, well, generate time course data for us because we need to parameterize your models. And that's exactly what I would have said if I had gone first, but I went last. And so every modeler on the panel said essentially the same thing. And at the end, just seeing the disappointment on this guy's face made me realize that this wasn't what the answer he was looking for. And that's because generating time course data is usually difficult. It's not the type of data that experimentalists generate routinely. And it's, expensive and just difficult to generate. But this is really kind of the best way to parameterize models. The models we tend to develop predict time courses, and so the natural input for parameterization would be time courses. And so this is just a slide showing an illustration of a model and the time course data that we use to parameterize it. In the literature, what we find more regularly is qualitative data, like what we have here. So we have strains of yeast that have temperature-sensitive mutations. So at a permissive temperature, they can all grow. But you can see here on the second row that at the higher temperature, mutation of CDC14 results in a growth arrest. That can be rescued, as indicated here, by overexpression of another protein, SIC1. And SIC1 is a cyclin-dependent kinase inhibitor, and CDC14 is a phosphatase. And what you can see is there's not numbers associated with this data, but it does tell us something about how the system behaves. So I'm going to start with our work by just showing that qualitative data can be useful for defining parameters. So we're going to consider this really simple case where we have a parabola and a line. And let's assume that we know 2 points on the parabola and 1 point on the line without error. There are 5 coefficients in these 2 polynomial equations, but the data point, the points on the curves, that's not adequate to determine any of these coefficients. But if we could compare the 2 curves, scoring whether or not the parabola is above or below the line, so here it's above, at this point it's below, at this point it's below, at this point it's above, and over here it's above. So if we had that qualitative information, that would allow us to determine a range of x values where there's crossing of the 2 curves. So here the parabola is above the line, here it's below, so we know that it crosses somewhere in between. And similarly, we know that there's a crossing point between this x value and this x value. And if we have just a little bit of this information, we can constrain the coefficients. And if we have a lot of this information, we can determine the coefficients exactly. So again, this is just a simple illustration that qualitative information can tell you something about numerical parameter values. All right, now I'm just going to cut to the chase and tell you what our approach is for automating the Tyson way of parameterizing models. So we take the qualitative observations and we convert them to mathematical form by writing the information as an inequality. So we have a function or a functional of parameter values of a model, and each one can be written as something less than 0. And I'll give examples of this later. So if you're curious about how this works, just wait, I'll give some examples and hopefully it'll be clear. One important point to note is that this is not a box constraint on parameter values. So it's very common for us in estimating parameters to say a parameter is between some lower bound and upper bound. So P might be between 10 and 100. That's a realistic range for this parameter. But that's not necessarily, that could be what this is, but it's not necessarily what it is. More generally, it's a constraint on the behavior that's predicted, not a parameter of the model. And then we solve a constrained optimization problem. So we minimize some objective function, trying to satisfy these inequalities through some approach. And there are many approaches. This is something that's really studied in the engineering literature quite a bit. And there's lots of ideas about how to solve constrained optimization problems. And one of the oldest ideas is to solve these problems with the static penalty function approach. And that's the approach that we took. So in the static penalty function approach, you introduce a penalty whenever there's violation of a constraint. So if this constraint is satisfied, G is less than 0. And so if you take the maximum of 0 or the value of the function or functional, you see that this term here evaluates 0, no penalty if the constraint is satisfied. And then there is a penalty that's proportional to constraint violation. And the penalty is determined partly by a weight. And that weight is a constant. And with this approach, it's assumed that the modeler knows how to set these constants. In more sophisticated approaches, in the optimization, these weights are adjusted as part of the procedure. But we're taking kind of the simpler approach where we can set these. It's assumed that we set these. Now, there's some points I want to emphasize about our approach. Again, the constraints apply to outputs of the model, predicted behaviors in general. The constraints are soft. So in some forms of constrained optimization, the constraints are required to be satisfied. So the method gives a solution where all the constraints are satisfied. We don't want that type of approach because our constraints are determined by observational data, and we may not trust all of the data equally. So there could be measurement error. There could be experiments done in a different system than the one we're interested in that should be relevant but may not be. And then there's this issue that's commonly that's well known. Different laboratories sometimes predict contradictory results. And so if we just take an unbiased approach and consider all the data available, we may have constraints that can't be satisfied at the same time. The other point that's important to note is that the penalties that we're introducing reflect the distance from the feasible region of parameter space. And that's important because this allows the optimization algorithm that you're using to assess whether a fit is moving toward or away from the feasible region. Now, in some ways of solving constrained optimization problems, you just count the number of times a constraint is satisfied, but that doesn't tell you how close you are to the feasible region. So we didn't take that approach, and I think that was important in our success. Having a way of solving this problem that allows the optimization algorithm being used to determine whether you're moving toward or away from the feasible region. And then weights are static, meaning they're constant, and they have to be selected by us. And there's no way to do this that's principled. So it's an ad hoc approach. And the rationale for selecting them is you want balance. You want all your constraints to receive roughly equal weight. So I have a little example here at the bottom to explain what that means. So let's say I have a function G1, and it's supposed to be less than 10, and I have another one that's supposed to be less than 100. I'd write those constraints like this, and then I would pick these weights. So I'd set C1 to be 1/10 and C2 to be 100th. And then in my objective function, if G1 is 20, so twice the upper bound, or if G2 is twice the upper bound, the penalty is 2 in each of those cases. So you can see how selecting these weights, lets the constraints receive kind of equal weight, if that makes sense. If there's any questions, maybe you can interrupt or ask me at the end. So I want to move on now to a simple example that's biologically relevant. So we looked at a model that Boris Holdenko developed to explain paradoxical activation of RAF. So we're considering BRAF, the mutant form, and a BRAF-specific kinase inhibitor, let's say vemurafenib. So RAF dimerizes. When it's monomeric, it's not active. When it's a dimer, it's active. And then this drug is interesting because it promotes dimerization. So if you have a saturating dose of the drug, then you're going to end up with all your RAF and dimers, and both copies will be bound to inhibitor, so there'll be no RAF activity. But if you don't have a saturating dose, you'll have significant population of this species where one copy is inhibited, but the other one isn't, and that other copy is active. So this gives paradoxical activation, which is shown here in the panel at right. So just showing what's happening as you increase the dose of the inhibitor, you have some baseline level of activity because of constitutive dimerization. The drug induces dimerization, so you get higher activity, and then at a saturating dose, you lose activity. So in this example, we generated data from the model based on assigned ground truth parameter values. And we generated quantitative data, shown here at the left, and qualitative data, shown here at the right. So the quantitative data has noise at it, Gaussian noise, we're just showing a fraction of RAF bound as a function of free inhibitor concentration. And this is data that could be readily generated. And then over here, we're showing, this curve shows RAF kinase activity, but we're assuming that we actually can't measure that quantitatively. We have an assay that just tells us how activity compares to control, so no drug, and we can score whether or not activity is within error of control, higher than control, or lower than control. So these symbols at the bottom are showing same as control, more than control, same as control, less than control. So we don't really know the extent to which RAF is activated. We just know that it's more activated than control. Hopefully that's clear. And we use and we can use either of these data sets to get good estimates for the parameters in the model. So I forget which curve is which here in these middle panels. One is the ground truth, the other is best fit. And we can get good fits using just the quantitative data or just the qualitative data, which is somewhat surprising. But what we're showing here at the bottom is that we do better at identifying the parameters when we use both data sets. So these are profile likelihood plots. If you're not, this is an old technique in statistics. What you do is you hold a parameter value constant and then you do fitting and you just plot your objective function at all these parameter values that you've just held constant here. And so lower value means better fit. And the way you would interpret this plot is, the data that we have is showing that, if this value is over here in this region, the fit's not good, but over here, no matter what the value is, so long as it's less than some threshold, fits are pretty good. So it's showing that the data is telling us the parameter is somewhere to the left at this point. And this red bar is showing the best fit parameter value. Then over here, we just repeat this for a different parameter in the model. And the purple curve or the magenta curve most tightly constrains the value of this parameter. So if we pick some arbitrary value for the objective function, let's say 0.2, then this curve is saying that to this level of confidence, the parameter is between this point and this point. And as you can see, the purple curve more tightly constrains our estimate than when we use the quantitative data only or the qualitative data only. So this is just a simple example illustrating how the method, the outcome of the method and how we assess whether or not it was helpful. Now, what we did in our studies, we moved on to a much more challenging example. We took one of the versions of the model for cell cycle control that John Tyson and co-workers have developed. It's described most fully in this PhD thesis. So this is just a diagram of the model. And this is an example of its predictions. So it's showing that certain proteins oscillate over time. Okay, so we wanted to estimate the parameter values of this model, not through hand tuning, but through an automated method where we're solving a constraint optimization problem. And we wanted to use the qualitative data that Tyson and coworkers originally used. So that's mostly, for the most part, viability status of 119 different mutant forms of regular baker's yeast. So this type of data. And then we added in time course data, and we just picked some data that was available in the literature, gene expression measurements as a function of time using microarray technology. So this is very noisy data. It was available, we picked it just to show that quantitative data could be considered if you wanted to do that, or if you could do that. So here's a slide illustrating the constrained optimization problem. So we have constraints on predicted behavior. So in this model, the phenotypes are viable or inviable. John Tyson and his coworkers have described what these properties are in like natural language or in programs that they've written. So they would sell, a property of a viable mutant is that it divides eventually. And so they set an arbitrary threshold of 8 on the volume of the cell. If the volume is always less than 8, then it's dividing, because in simulations, when there's division event, you follow just one of the two daughters, so the volume is going up and down. And if there's no division, then this volume grows, and 8 I don't think is particularly special. It's just a value that's large for this model. And as we talked about earlier, we add penalty terms to our objective function, and we can divide our objective function up into normal types of terms, like sum of squares, and then these penalty functions of the type that we talked about earlier. And then we can combine the two to get a single scalar valued function that we want to minimize. And that's how we, at this point, because it's a scalar valued objective function, you can apply any of the standard methods for unconstrained optimization. And, but it's difficult to calculate derivatives, so you don't want to use a gradient-based method. That's not really an option. But you can do something like simulated annealing. We used scatter search. Okay, so some things to note about this particular optimization problem. There's 119 mutants. These strains have knockouts of particular genes, and to simulate a knockout, we set the gene product abundance to 0. The behaviors of these mutant strains are formalized as about 1,600 inequality constraints. So each one has has multiple constraints, like that volume constraint that I talked about earlier. And then in the microarray data that was reported, we used 561 of the data points. These are the measurements that are relevant for the model. The model has, as in the original work, 153 free parameters. And then when we're evaluating this objective function, we're doing more than one simulation. Normally you would do one simulation, you get all the time courses, and you would calculate the distance between measured and predicted time courses, like a sum of squares function. But here, for each mutant, we have to set some gene product to 0 and then do a simulation, and then evaluate some number of inequalities. So each function evaluation is expensive. Again, we just use scatter search to solve it on the recommendation of Fred Glover. So this slide just shows some more examples of qualitative data. So I already talked about the constraint on volume, but over here you can see this is the behavior for a strain that is viable. This is the behavior for a strain that isn't viable. There's no division, so the volume never drops by a half. The model has some flags that are used to track certain processes that aren't, modeled in detail. So like spindle assembly. So there's a function in the model or a variable in the model, SPN, that is 0 immediately after division. And then if something else happens, if there's a trigger event in the model, it starts to increase, and spindle assembly is taken to be complete when it crosses a threshold value, which is 1. And so this is the mathematical form of that observation. And it just says that at some time, this variable has to be greater than 1. And so here's a behavior that's consistent. You can see that spindle is exceeding the threshold value repeatedly. And then here's a version of the model that corresponds to some mutant where spindle assembly doesn't complete. And then we took into account not only viability and inviability, but if we had an inviable mutant, we considered where it was arrested in the cell cycle. So if there's a mutant that's arrested in the G1 phase, that translates in natural language to this flag ORI in the model does not reach the threshold value that signals completion of origin activation. And then in mathematics, that translates into this constraint, which says that ORI must be below, remember, this is an inviable form of cell, must be less than some threshold value during the period during which the volume hasn't yet crossed the threshold. So here's what a simulation looks like for a version of the model where you do have G1 arrest. So the volume increases without bound, and during that period, this flag never crosses the threshold value. So it's arrested in that phase of the cell cycle. And here's what a simulation looks like where you don't have that behavior. So there's 1,600 of these things, which Ishan wrote by hand. That's what you'd have to do to account for qualitative, any sort of qualitative data. You'd have to write a mathematical inequality for each observation. And this slide shows the quality of the fit that we achieved. So in fitting, we use both the qualitative data and the time course data. And these are simulations showing consistency with some of the qualitative constraints. And over here on the right side, we have time courses, and the turquoise curve or the cyan curve shows the behavior that our parameterization predicts, and then this orange curve, dashed curve, shows the prediction of the simulations predicted by the original parameterization by the Tyson group. And they do okay, but we do a little bit better because we use the quantitative data. They didn't use the quantitative data. And then we assessed how well we did using profile likelihood analysis. So I'll just focus in on this upper left plot. So this is one particular parameter in the model. Again, we fixed it at these values along the x-axis, repeated fitting at each point. And if we use only the quantitative data, this flat curve indicates that we don't know what the parameter value is. All parameter values are equally good. Whereas if we use only the qualitative data, we see that, let's say we're going to pick 100 as our confidence level, then the value is between this point and this point. But if we use both the quantitative and quantitative data, we're able to identify a more narrow range at the same confidence level. And that's the trend. So by using both types of data, we get reduced parameter uncertainty. And then the main point is in solving, in parameterizing the model. That was already done by the Tyson group. The contribution here is we showed how to systematize the procedure, make it reproducible and automatic. And so what Ishan has done since this proof of principle demonstration, which was accomplished with problem-specific code, is he's implemented the procedure in a new version of PieBarnettFit, which we're calling PieBarnettFit. Source code's available here. The way PieBarnettFit is designed to work is you define your model using BinetGen language or BNGL or SBML. You then define the data that you want to use in fitting, so you can have time course data, or you can use a system property definition language that PieBarnettFit understands. So here's two examples of constraint of the type that we've talked about. So this is the v is less than 8 always. Here's another one, y is greater than 1 at time 10. So it's a very, I mean, we've been able to express a lot of qualitative properties using this constraint language. It might need to be extended in the future to account for things that we haven't thought about yet. And then you provide the software a configuration file which tells it, you know, which optimization algorithm you want to use. There are several that are offered, and then for the one that you pick, you have to sometimes define algorithmic parameter settings. So that's done in this configuration file. You then solve the optimization problem, and you get best fit parameter values. So if anyone's interested in trying this out, it should be easy to do with our software. At this link, you'll find all the source code and documentation as well. There's an extensive user manual. So this is the end, and I want to emphasize a few points. So again, our contribution isn't in solving the parameterization problem for the cell cycle model. It's in showing that the procedure can be automated. You know, as I mentioned, there's general purpose software available now. It's compatible with SBML and BNGL formatted models. It's integrated with various simulation engines, those of BinetGen, NFSim, and the libRoadrunner library from Herbert. The software includes a system property definition language, which can be used to define these constraints on predicted behaviors. There's a variety of optimization methods, and for the most part, these methods are able to leverage parallel computing resources, which, given the expense of these optimization problems, is very important. So we're now using or contemplating using millions of CPU hours for fitting problems that we have. You know, one intriguing opportunity now is that maybe it's possible to design high throughput experiments that could generate loads of qualitative data that could be useful for parameterizing models. So maybe there are new ways to parameterize the models that we want to develop that are open to us now that weren't before without automation of the Tyson approach. And I also want to talk about a couple of things that we're thinking about. So just sharing our ideas about where we could go with this. So one future direction is just taking this property definition language that PieBarnettFit has and using it to define the behaviors that we think a model should reproduce, and then taking the model that we're working on and seeing if it actually does reproduce that. If we have multiple models for the same system, maybe we can take a library or database of system properties and then compare them. So we might be able to say, oh, I've got models A, B, and C reproduces way more properties than the other models do. So it's the one that I want to pick up and refine as I move forward with my modeling. And then, you know, another thing that I think is possible is design. So we can take the system property language, use it to define behaviors that we want a network to have. And then we can take the optimization capabilities of PieBarnettFit and use them to find perturbations that yield the desired behavior. So that might be achieved by, knocking out specific sites and proteins considered in a model, or knocking down expression of particular genes by, like, say, tenfold, or just knocking out expression. And then we can consider reductions in enzymatic activities achieved through molecularly targeted drugs, for example. So I think that's a really intriguing possibility now. And of course, this has always been possible through ad hoc analysis, which I would say is the standard approach at present. But I think, with a systematic automated approach, it's going to be less likely that we miss interesting opportunities for interventions. So that's the end of my talk. I hope it's been interesting and we have a good discussion. Thank you.
Thank you so much for your talk. This software really sounds incredible.
It looks like I went over by, I'm sorry about that. You should have stopped me.
It's totally fine. And this is like, I've never seen anything that is this powerful before, so I'm extremely impressed. Let's start the discussion. First off, Bill, would you be able to close your share screen?
Oh, yeah, absolutely. Let me figure out how to do that. How do I do that, Ishan? Where do I go to do that?
So if you go to like share at the bottom of the screen, you should be able to
stop sharing. I see it. Yeah.
Okay, so to introduce the discussion topic, first off, just two quick announcements, and then we'll get to the discussion. First is that for whoever is willing, we would like to host your bio and your name on our website to sort of consolidate our community, and I'll send an email about that afterwards. Also, if anybody is free, for approximately 10 to 20 minutes after the seminar at 4 o'clock, we're going to continue to do introductions if anyone would like to introduce himself and hasn't yet. Okay, that being said, I think this would be a good opportunity to discuss what types of data are available for calibration and where we can get it. And then also, which sorts of data would be necessary. So does anyone have any thoughts on where we can get data and how it can be implemented in calibrating models?
There are some text questions too if you look in the chat.
True. Oh. That sounds good. Yeah. Let's start with that. Okay. So first we have a question from Herbert Sarah. I guess Dr. Sarah, would you like to read your question?
That's really a technical question, and I'm glad I have Ishan here to answer it. You want to get into that, Ishan?
Right. The question is, how many optimizations do you do to compute the profile likelihood plot per parameter?
Yeah.
And so I guess the answer is it depends on how densely you want to sample that curve. The last plot that Bill showed had individual data points on it. I think it was about 10 data points. And that's because that was an expensive optimization. And so we could really only afford about 10 times. So it's one optimization per data point. In the earlier, simpler model, that was not an expensive optimization. And so we were able to sample it very densely.
How densely?
The resolution of that figure, basically.
Yeah.
So a lot of data points.
Yes.
I'm going to close this.
That one's muted.
I need to mute.
Okay, yeah, we had a feedback. Oh, it's gone now. So I was curious about how it scales compared to, Monte Carlo estimation, where you sample the data and rerun optimization. It sounds like profile likelihood is less demanding.
Yes, I'd say that it's a lower total number of evaluations than Monte Carlo approach.
Yeah.
It doesn't give you quite as much information as a Monte Carlo approach because that would allow you to sample the entire probability distribution and identify correlations between parameters, whereas profile likelihood is only looking at the identifiability of the single parameter that you are fixing.
I see, yeah. Okay, yeah, okay, thanks for that answer, yeah, that's good.
All right, and next we have a question from Gary Ann. Gary Ann, would you like to read your question?
Yeah, actually, I think that last answer answered that question because I'm more interested in creating probability distributions of feasible parameter sets as opposed to finding whether a particular parameter set is feasible. So I think that answers the question, if I understood the answer to the last question correct.
Yeah.
Sounds good. Yeah, sir. Would you like to read your question? Okay, well, I'll read the question. Does the method work for higher dimensional problems? I guess, how many parameters?
Yeah, I mean, because the evaluation of the objective function is expensive, it's going to work better when you have fewer free parameters, I would say. But I mean, we solved a problem with 153 free parameters. That's pretty high dimensional. Yeah, I mean, if you have more parameters than that, I guess you're getting the trouble you're asking for. But no, it works. I would say that was a hard problem.
So Bill, what machine did you use to run that problem? Are you using desktop machine still, or you're running on cluster?
I'll let Ishan answer.
Yeah, that particular problem instance was on a multi-core, 24-core desktop machine.
Okay.
And I see there was a question about how long it took. It was on the order of 10 days or so on that machine.
Yeah, okay.
But that's to be compared with, the effort to hand tune the parameter values.
Yeah, and you only have to do it once anyway.
Right.
So, okay, good.
Can I follow up? My question, you sort of answered it, but I wanted to know more specifically. You might have said something a little bit earlier, but about how many simulations can you do, like in a minute or a second, on one of these machines?
So, what the cost is will depend on your problem. And one of the nice features of PieBarnettFit is that the algorithms that are implemented in it, for the most part, not all of them, but for the most part, can take advantage of a cluster. So if you have access to a very large cluster, you can solve like super hard problems.
Okay, yeah, I understand that, but I'm really interested in how long it took you for the Tyson model, because I have experience with that model and I've simulators for it too.
Because that is a differential equation model that's numerically integrated, it's quite fast. The number that I recall, it was on the order of 1 billion individual simulations over that 10-day runtime.
Cool. Thanks.
So that was parallelized on a 24-core machine, so you can calculate that out.
Yeah.
Cool. That's exactly what I want to know. Thanks.
I have a question about getting data for this calibration. So you mentioned that you had sort of made, I think, 1,600 different modules that it can be calibrated with. Which types of data is it calibrated on, and where is this accessible?
So the particular data set that we used for calibrating this proof of principle model was a data set compiled by John Tyson and coworkers. And what they did was search through the literature for experiments. Bill showed one of those experiments in his slides. Each experiment made a particular mutation to yeast and measured whether it was viable or not. And John Tyson's group tabulated all of that data, and that's what we used as the
So the data was the same data that they used, and nicely they shared the source code of a program, and I think that's where you actually figured out what was going on for the most part by looking at that code. And if you look in our supplemental material, we give all of the constraints, so they're all enumerated in the supplemental material. And I think that's one of the strengths of the approach, that, you know, there's no ambiguity about what we're fitting to.
Could I ask another question? In your talk, I can't recall the meta-heuristic approach that you used, but how easy would it be to implement, you know, some other meta-modeling optimization strategy like random forest, or you mentioned simulated annealing, right? Can you take off-the-shelf packages that do those things, or do they need to somehow be ported into this framework?
So if you want to use our software, you know, you're stuck with what we implemented. We did implement a variety of tools. There's a differential evolution algorithm. There's a particle swarm optimization algorithm. There's a scatter search algorithm. It also does Bayesian parameter estimation. So if you're interested in that, as I think you said earlier, that might be of interest. So that's just regular Markov chain Monte Carlo, and then there's a parallel tempering algorithm. Is there anything else, Ishan?
There's simulated annealing.
Simulated annealing, okay. Yeah, and you know, the reason we provide a toolbox is because for a given problem, you know, a given algorithm is not going to perform well. So it's good to have options. And what we're seeing in our benchmarking is that, you know, for a given problem, one of the methods will do better than the others, and that method changes from problem to problem. Is that right?
Yeah.
Yeah.
Hi, I have a question. Actually, it's posted there. Can the method handle or take input dynamic features as constraints? For example, if you have some observations that under certain perturbations, the system is oscillating.
Yes. So the G's, those can actually be functionals. So that means it's a function of the behavior over time. And that's what you're describing, I think. So the answer is yes. How exactly you would describe oscillations, though, I don't know if that's a built-in feature. We have a built-in feature that allows for that.
We do not have specifically oscillations support, but
yeah. So that might be added to desired features at some point.
Yeah.
Okay.
Yeah.
Thanks.
I have a question about scalability. How does it scale in terms of number of variables and number of constraints? The computational cost. Is there any nonlinearity?
We evaluated that. Like, I don't.
So in terms of number of variables, but of course the general rule is that it gets higher, but it with more variables. Just
for an ODE model, it goes with like a power of 3, the number of equations.
Right. So not just the simulation cost, but the number of parameter sets that you have to try with one of these meta-heuristic optimization algorithms.
But I mean, it's roughly proportional to the number of constraints, right? Because you have to do a simulation for each mutant, right? So that's roughly
well, so it's definitely proportional to the number of mutants. If you had a lot of constraints on
right. So if there's one mutant that required 100 constraints,
if you have a single simulation that you're running that had 100 constraints, I don't think that would be much worse than a single simulation and 10 constraints.
Yeah, right.
Unless it somehow made your parameter search space more irregular and cause
constraint evaluation is probably cheap.
Constraint evaluation is definitely cheap. It's the simulation is the main cost.
Yeah.
And coming back to Rudy's question, which I had seconded in the chat box. I think that's a very important point. There's additional features that come to mind in terms of dynamic features like, slopes and switching. That would be really nice if you could define them as mathematical constraints. If you had thought of any other ones, obviously oscillations is a very common one, but there's more dynamical features that are relevant qualitative fundamental differences.
So just a general comment. I mean, we would love feedback about what should be in the system property definition language. It's described in the user manual. Is that where you should look for it? Usually?
Yes.
Yeah. So it's described. You can see what's there. And if you guys want new features, yeah, we'll put them in. I mean, what's in there right now is kind of what's needed to solve the Tyson problem. So, and it was initially solved with problem-specific code, and then we built the language out to allow us to solve that problem again with the general purpose tool. So, we don't have a lot of experience using it, and so it's probably incomplete.
On the topic of the systems verification language, I was wondering if you could compare this to formal verification or unit testing as other potential approaches to verifying the behavior of models.
Yeah, so there are people that have been inspired by model checking in computer science, and this is related. In all the work that I'm familiar with, you use temporal logic, like there's abbreviations for these things like LTL, linear, temporal. There's different temporal logic. So basically just different languages for defining properties. But because in computer science, there's no such thing as a mutant form of your computer. It's useless. It's famously used to verify hardware. And there aren't mutant computers. So there's no really allowance in the computer science literature for a case control comparison, but we have that all the time in biology. And so in the papers where people have been applying model checking ideas from computer science to biology, it's like, they define, I want this value to go above 3 or something. Whereas, and maybe this is something I didn't emphasize, but it's a very important point. In the PieBarnettFit property definition language, you can say this variable has to go above the value predicted by a different model, right? So you can do these case control comparisons. So almost everything in biology is measured relative to control. So you're kind of comparing two things, or simulate. You'd be comparing results from two different simulations, and that's allowed for. And that's a very important difference between what I know about model checking in biology and what we're providing. Didn't I say that right, Isha?
Yeah.
Okay. All right.
I have a question about your constraint language thing. Can you imagine, as you moving forward, standardizing that in some form, like either as a form of like SBML constraints or as SetML, some form like a constraint in SetML itself?
I think we've talked about that with Herbert, and yeah, I think that's a direction that we're interested in moving, and having input from you guys would be super helpful. I like domain-specific languages that are human readable, more so than like a.
Yeah, you definitely want a human readable form of it, but like a human readable and a computer readable form is usually nice for all of these things. Yeah.
C or C++ is human readable. Well, some may debate that, but it's human readable and computer readable, so you get the best of both worlds.
But not, but I would say domain specific. So, kind of as simple as possible.
No, I'm fully on board with you on that. Yeah, no, I agree with you. Yeah, definitely.
Do you think you can get by with just a declarative language, domain-specific language for such optimization, or do you need it to actually outline the steps?
Good question.
I don't know.
A question on the modeling. When you have a complicated model like you're working on, is it ever helpful to add noise to each of the data points in a constrained way, and explore the different configurations of parameter estimates that you get?
I don't think, would noise be, I don't have an opinion about that, but I would guess that noise is always bad. You always want to have perfect measurement, but that's not realistic. So in our, so in the data that we used, the time course data, that was definitely noisy. And in the case where we used that RAF model to generate synthetic data, we added noise.
Well, what I was thinking was that if you get different correlation structure,
I see. Okay.
Adding different noise to each of the data points, that might be informative on the efficacy of the network configuration.
So that is more sophisticated than what we've been thinking about, trying to take advantage of correlations. We don't really have a solution for that.
Sounds like it would take a long time to do that, though, on your model.
So we're close to the hour.
So there's another question on the chat list.
Hi, John.
Joseph, you want to read that?
Yeah, that sounds good. I guess this will be our last question. This is from Leonard. In your examples, you had to define various thresholds and constants to define the objective functions for qualitative data. Have you investigated the sensitivity of your results to the values of those parameters?
That's a good question.
The short answer is no. And the uncertainty quantification, that depends on the settings for those weights. So because that determines what the objective function value is. There is no, for the approach that we described, there's no principled way to define what those weights should be. It's really on the user to select them judiciously. Now, in the optimization literature, there are more sophisticated algorithms where those weights are found as part of the optimization. And so at some point in the future, we could implement one of those methods and see how it does. But we took the simplest approach here in our first proof of principle demonstration.
All right, and with that, I think we'll wrap up. Yishan and Bill, thank you so much for presenting this work.
Thank you for the interesting discussion.
Of course. We hope everyone will join us again in a month for the continuation of the seminar. And for anyone who has time, to proceed trying to have us form a community, we'll continue with introductions for those who haven't gone. So thank you.
All right. I think we have to